Hello friends, so welcome to the lecture 2, part 2 of the week 2. So basically in the past lectures we have looked at the short read and one of the long read sequencing technology. So in this lecture we will slightly brush up those concepts and also move forward with the another long read sequencing technology and a new technology which has come by Illumina for the link read sequencing. So basically Illumina, we have discussed that it is very good for short read sequencing, where basically you have to break the DNA into smaller pieces, right, and then prepare a library, go for the amplification on the flow cell, right, and then each of these amplified cluster, right, is the cycles of the sequencing happens there. And basically we are following the sequencing by synthesis principle here. So basically it needs an optical system because a camera has to record the fluorescence, okay, and there are 4 types of different fluorescence ATGC, but these days they are also like using only 2 of these fluorescent dyes and basically with a modified software, right. Read length is like not very large, up to 300 base pair, and okay, so basically this is the kind of image that they are capturing and then they are calling the bases. And so good thing is that it is very high throughput, massively parallel, very low cost, and basically okay, so that is why it is kind of a gold standard for the sequencing. But the problem it faces is that it requires a lot of assembly issues and software which work on this short reads and assemble it further, okay. Then we also looked at the link read sequencing, where the fragments of DNA, when they are generated, okay, they are labeled with different barcodes, and later on when they are sequenced, even the fragments are labeled with the same kind of barcode, and later on they are combined together into the sequence or the fragment from which they were actually derived, okay. So this kind of helps in building a very contiguous or long assembly. So basically this is offered by 10x Genomics, and the sequencing will be done, so basically library will be prepared by 10x Genomics instrument Chromium, and then the library will be sequencing will be done by any of the Illumina instrument, okay. So this was a good option to assemble large genomes. Then we looked at the another long read sequencing technology, which is very friendly to smaller labs, and you can sequence larger genomes, which is Nanopore sequencing technology, where basically there is a you can see here a nanopore from which the DNA passes through, and so basically since DNA is passing through a pore and it is disturbing the current that is earlier built up by the flow of ions. So the basis of the disturbance of current, or basically the voltage, okay, determines that what base pair is actually scanned through. So as you see that it will not need any nucleotides or any sequencing by synthesis or chain termination method, but just detection of base by base, okay. And and that is why it can produce very long fragments up to like hundreds of kb, and an average read length is also 10 kb, which is very good for larger genomes assembly. Although it suffers from some errors and it does not you need a second check for the sequence quality. So either you do that this sequencing in more depth, or you can combine the Illumina high quality data with this and improve the sequence quality of the nanopore assembly, okay. So various instruments like MinION, GridION and Prometheon are available. Library preparation is not difficult, it's quick, basically few hours thing, right. And yeah, so this is one of the latest things which is going on all over the world, okay. So the advantages and disadvantages we have discussed. So shorter reads we are sequencing. So imagine this is a picture, okay, you want to assemble this picture using a very short of these pieces, so you will require more effort, okay, logic, and also sometimes you are not sure which piece goes where, but if the piece size is bigger, or you can say the read length is bigger, you can always assemble it better, easier, and with higher accuracy. So that is why longer reads, link reads, these have a better future. Okay, so we have seen Nanopore, but now we should see another link long read sequencing technology, which is also very popular, although it is a bit expensive, and not all research groups and labs can afford the instrument or the sequencing, but this is also improving and basically used for larger genome sequencing. Okay, so for example, so here they have used a very beautiful approach where, so basically this is a piece of DNA. Okay, it has two strands on both sides. So what they have done is they have attached a bell-like template or hairpins kind of things on the both the sides. Okay, and then basically the DNA. So okay, so each of this piece, which is the smart bell SMRT template. Okay, SMRT means single molecule real time sequencing. Okay, so each of this piece goes into a ZMW well where actually the sequencing takes place. Okay, so you can see that each piece is going into a well where it is being read by the polymerase. So this way you can, so it is also called rolling circle replication for concept it follows. Okay, so this way basically the template fragments are processed and ligated to a hairpin structure. Okay, and this the single stranded molecule, the polymerase moves across that molecule. Okay, and with the help of a primer DNA polymerase you can sequence it. So it can also, it generates very long sequencing reads, and for example per SMRT cell you can get almost like earlier these days up to 500 GB of data with 2.0 chemistry, and so basically this is one of the important system these days to generate the longer reads. The error rate is also decreasing, although it is also similar to the Nanopore in error wise, but again the hybrid assembly approach works here. Okay, so basically here the sequencing is done in a ZMW, okay, which is inside that space where actually that hairpin structure went. Okay, so basically that single molecule DNA is immobilized on this, and the light is used for detecting each and every base pair that is being incorporated. Okay, so this is also known as PacBio sequencer. Okay, and the concept is SMRT sequencing. Okay, so you can see here that basically this is a one SMRT well and bell kind of structure at both sides. Okay, and which opens up and the polymerase moves, and the DNA strand, single stranded DNA is now made into another strand by replication, and while it is happening, basically the fluorescent light is being generated, which is read by the inside the ZMW. Right, so basically it produces long reads, very good for genome assembly, for transcriptome, because in transcriptome you want to know that what RNA sequences are there. So if you get the complete intact RNA, you will know about the alternate splicing and their copy number also very well. In epigenetics, because it also captures the methylation pattern of the DNA. Okay, it gives a uniform coverage across the genome, so it will not suffer from the bias that may happen during the library preparation. Okay, accuracy is being increasing, and it's a like single molecule resolution. So this is one of the better systems these days. Okay, so just to compare basically the SMRT, PacBio and ONT, you can see that this is happening inside the ZMWs. Okay, so because it is using polymerase and nucleotides, so it will be expensive, and that is why the instrument is also expensive, and also the consumables and library preparation. Whereas Nanopore, because it is not using a polymerase and the nucleotides, it is a cheaper or economical system, because like just the signal is being detected by the change in the flow of ions, right? So but both of these technologies are nowadays used very commonly, and are the gold standard for the long read sequencing. There is another link read sequencing technology which is okay, offered by Illumina. Okay, so in this basically you can see that there is a bead on which they try to capture the DNA molecule, okay, and using the transposons they break the DNA molecule. So it is transposase enzyme linked long read sequencing, that is why it is called. Okay, then for each piece they will put their adapters and amplify them, and okay, so basically the strength of this link read technology is that although the principle is similar to the other link read technology also, but the strength is that it can work with ultra low quantities of DNA. Okay, so basically you have very low amount, 0.1 to 5 nanogram of DNA, you just barcode it, okay, and then basically amplify and then go for sequencing. So within 3 hours you will get the data. And they have also developed their own sequencer called Turing assembler called Turing assembler, which is good for the assembly of these reads, because you have to assemble the link reads, right? So this is the TELL-Seq technology which is offered by Illumina. Okay, so and basically after the library preparation, the sequencing is done by any of the Illumina platform. It could be Mini-Seq or MySeq or NextSeq, right? The larger ones may not be appropriate because they can do much more than that, but I think any smaller or medium level system is okay to assemble these. Okay, and yeah, you can prepare with very low input amount, whereas PacBio and Nanopore needs larger amounts of DNA. Okay, so in the end I will give you a quick illustration of how this DNA sequencing thing will happen for a real-time project, right? So let's see like how, for example, a metagenomic project, how can we use these sequencers. So what I have showing here is a NextSeq 500 sequencer, okay, which is a medium level medium output sequencer which produces about 40 to 120 GB of data. Okay, it's a desktop instrument, very small. Okay, just plug and play cartridge and flow cell you can put, and it is not difficult to operate also. Okay, good for genome, transcriptome, amplicon sequencing. So it offers, so okay, so like this is the sequencer, okay, this is the flow cell, right? Different types of outputs you can select during the library preparation and the flow cell, right? And these are the various consumables. This is the cartridge for the various nucleotides and the other consumables which are required for sequencing, right? And the buffer cartridge. And it offers different types of libraries such as Nextera, XT-DNA, okay, which is where the starting material is 1 nanogram, and okay, up to smaller genomes it can do, because you are taking smaller amount of DNA, so you will not get okay, that much diversity in the read. So for smaller genome it is very good. Then basically another one is Nextera DNA Flex, which is starting material up to 500 nanogram, and little larger genomes can also be done. But for the longer or larger genomes we use TrueSeq technology. So there are two variants of that again, TrueSeq DNA and TrueSeq Nano, okay, where basically in DNA it is the starting material about 1 microgram, and but one safer side you take more, okay, and then keep it reserved for later analysis and later things, okay. And for the TrueSeq Nano it can work with up to 200 nanogram DNA also. So these two are for the larger genomes, and Nextera is generally for the smaller genomes, okay. So now basically there are two types of metagenomic approaches. So one where you are only interested in reading or identifying the marker, okay, so you will just amplify the 16S ribosomal RNA gene, the complete one or the variable region, okay, using a set of primers along with the barcode, okay, and then you can know that after the assembly you can know that what is the taxonomic composition of the sample. And the other one is the where you want to sequence each and every bacteria in the environment in their respective proportion or abundance, okay. So basically it is you are fragmenting all the DNA which is present from all the bacteria in their respective numbers in an environment, okay, and then preparing a library, going for sequencing, and then assembly and data analysis. Okay, so in the amplicon base, you are trying to identify only what is there, like what organisms are there in metagenome, whereas in the metagenomic approach, you are trying to you are also getting access to the entire genetic material, genes, and including the markers in the metagenomic DNA. Okay, so basically you will be collecting the sample will reach to a lab, you will store it properly in the lab. Either you process it immediately or you have to store in appropriate conditions, right? Preferably -20 for shorter storage and -80 for longer, right? And after that you will do the metagenomic DNA extraction using a proprietary kit. There are several versions available for the different type of tissues that you are going to handle. Okay, and then going for the library preparation using the Illumina provided consumables for library preparation, like 4 library preparation methods I just told in the previous slide. Okay, okay, and then you can also do indexing for samples, just in case if you are multiplexing or putting too many samples together, right? Because you have to accommodate 120 GB of samples, okay, because that is the output of the sequencer, and for each sample maybe you are looking at 5 GB, so you can put like 120 divided by 5, that many number of samples in that. So for that you may require multiplexing, going for sequencing, and whatever data comes going for data analysis, right? So this is a typical workflow in which so right now I'm telling about the metagenome. Similarly for a genome, you take the sample, okay, store it, store the DNA, extract the DNA, and then going for the library preparation and the sequencing. So okay, so basically we'll be covering the sequencing application aspects and how to approach the assembly, the genome sequencing and assembly, and how actually to design a genomic sequencing project in the later part of our talks. So but for this class we covered one more method of long read sequencing, that is SMRT or the PacBio sequencing. Another link read sequencing technology which is called TELL-Seq, offered by Illumina, and an example of a metagenomic study where from sequence we are generating sequence data from the sample that we have collected or any of the project. Okay, so this is good for today, and we will see again in the next class. Thank you.